So, let's get going. This is the last lecture, and today we're going to talk about apoptosis, or programmed cell death. It's something that is kind of personal to me. That there is my left foot, and when I was born, my mother noticed right away there was something a little odd about my toes. So my second and third toe are webbed and joined together. And so what had happened while I was in my mother's womb is that a developmental process didn't happen quite properly. There's probably a couple of other developmental processes that didn't happen properly, but let's not focus on those. So normally what would happen here is during development, the digits, the fingers and your toes, they're kind of formed like little clubs. And then there's a selective death of cells called interdigitization. And so that death of cells occurs through a process called apoptosis. And obviously that didn't happen with me. And yes, I am a good swimmer. Okay. So today we're going to talk about what is apoptosis, what distinguishes this from other forms of cell death, how did we figure it out. A little worm called C. elegans played a big role. We'll talk about some of the major players. We'll talk about, there's a bit of a constant battle between proteins that turn on apoptosis and the other ones that try to stop it. And we'll talk about apoptosis can be actually activated within, usually through the removal of certain survival factors, or extrinsic factors can actually trigger the death of cells. So, apoptosis or programmed cell death, it is a normal process. It's obviously, as I talked about, plays a role in normal embryonic development. It also plays a role in your normal tissues. When they kind of reach the end of their life cycle, they usually die by apoptosis, and they can then potentially be replaced by stem cells. And one key thing to note about apoptosis is that it actually is a very coordinated series of events. It's like the controlled destruction of the cell. When you think about when you're going to demolish a building, and you want to minimize the impact of that building when it's being destroyed. You don't just put a big bomb in there and blow it up and it goes everywhere and causes destruction. You selectively plan detonations that weaken the structure and cause it to self-collapse. It's a little bit like that with apoptosis. It goes through a series of events where the cell actually starts to shrink a little bit, the chromatin becomes more condensed, and then what happens is the nuclei start to break up, and actually the cells start to break up. And so you get chromatin condensation, you start to get the cells shrinking a little bit, and then you get this blebbing of the cells. And then eventually what happens is you get these things called apoptotic bodies. So it's like little pieces of the cell held in baggies. So you kind of encapsulate the guts of the cell in these small apoptotic bodies. And so the apoptotic bodies then can be recognized by phagocytic cells and degrade them. So the key aspect here is that by not causing the non-specific just rupture of the cells, which could trigger an unhealthy immune response, you maintain all the intracellular contents as these apoptotic bodies so that then they can be recognized by sort of clearance cells, phagocytic cells, and immune cells. We'll talk a little bit more about that later. This would be opposed to necrosis, which is usually where cells swell up and burst. And that can still happen under extreme stress conditions. But apoptosis is actually a coordinated series of events. The cell actually activates signaling pathways. In some cases, it activates certain gene expression pathways to actually cause the destruction, its own demise. Another thing that happens during apoptosis is that the DNA, there's DNases that get activated, and they cleave internucleosomal regions of DNA. What does that mean? Well, the DNA is wrapped around histones or nucleosomes, and there's a periodicity of that, about every 200 base pairs. About 200 base pairs wrap around the nucleosome, and then there's the space in between the nucleosomes is where the DNA cuts. And so it cuts at roughly 200 base pair intervals. So if you take genomic DNA from an apoptotic cell and run it out on an agarose gel and stain for the DNA, you see this DNA ladder. This is actually just from my PhD thesis from a long time ago, where in this case it was trophic factor withdrawal withdrew a cytokine called interleukin-3, and it led to the apoptosis of these cells. Okay, so a little bit more about some of the differences. So as I said, necrosis is generally the cells tend to swell and they rupture, and this can be bad because you're releasing intracellular proteins which can trigger an inflammatory response, which is not good. By compartmentalizing the cell and breaking it down into pieces into these apoptotic bodies, they can then be recognized by phagocytic cells. And one of the ways that this happens is that there's a restructuring of the plasma membrane. I won't get into all the details of it, but basically there is a phospholipid called phosphatidylserine, and that's normally facing, it's at the plasma membrane, but the side that's facing to the cytosol, or the cytosolic leaflet of the plasma membrane. And during apoptosis, there's some stuff that goes on, which I'm not going to test you about, but it kind of flips these phosphatidylserines from the inner part of the cell, and they flip to the outside, to the outer membrane of the plasma membrane. And this acts as a eat me signal because there's receptors found on phagocytic cells that will recognize these phosphatidylserine phospholipids, and that promotes their phagocytosis. So, apoptosis was probably identified about 50-ish years ago, and people at first were kind of skeptical, thinking that does this really have much relevance, and how would we ever understand such a process? And it turned out that this lowly little beast called C. elegans is what helped really identify the molecular processes that control apoptosis. And it's only a couple of millimeters, or maybe 1 to 2, maybe a big one might be 2 millimeters long, barely visible to the eye. But it has some properties that are convenient when you're doing research. One is that it's transparent, so you can see inside, you can see cells inside the animal. It has a defined number of cells. So there's two sexes, there's a male and then hermaphrodite, which has both male and female organs. And roughly, depending on which sex, about 1,000 cells, give or take. And so we know all the cells in the body, they're all mapped out. The genome is fully sequenced. It's very easy to make mutations in these animals, and you can actually silence gene expression by feeding them bacteria that encode small interfering RNA. So this actually can silence gene expression. So it was a very versatile organism to understand cellular processes, and these fellows ended up winning a Nobel Prize in this 20-odd years ago, related to the genetic regulation of not only development, but also programmed cell death. So it turns out that C. elegans goes through some developmental processes, and just like my foot, when it was developing in the womb, you get some extra cells, and then you kind of have to curve them back. So if we look at the sort of the later stages of development of the worm, before it actually matures into an adult worm, it has an excess of newborn cells. About just over 100 cells die during development, so you end up with slightly less than 1,000 cells. And as these cells die, you can see them depending on the type of microscopy. In this case, it's DIC, or differential interference contrast microscopy, which allows you to see certain features in the cell. So this is using light microscopy with some contrast enhancement through this DIC process. And you can see these apoptotic, or these dead cells, because they're more refractile. They kind of appear as blebs, so to speak, inside the animal. And so researchers wanted to see, okay, we know that the worm makes more cells than it needs before it finally matures. It has to get rid of those cells. We can see those cells inside the animal just by a simple light microscopy. So we can maybe mutagenize these animals, give them small, very low levels of a mutagenic agent, and then look for, do we get more dead cells or do we get less dead cells by just simply looking at them. And that's what they did. And in many cases, well, some of the first mutations that they identified were in genes that
which they called SED genes, stands for cell death abnormal. And so the first two genes identified were SED3 and SED4. Normally I want to school people for turning on. Sorry, just a second. There we go. So SED3 and SED4, if they're mutated, you get you don't get so those dead cells, you don't see them as many. So you're reducing the number of dead cells. And then they also screened for other mutations. In this case, a gene called SED9, when mutated, they get massive cell death, or apoptosis during embryogenesis. So what that suggested is that these genes, if they're functioning, not mutated, actually promote cell death. If they are mutated, i.e. they do not function, then you're not getting a cell death that would be part of the normal developmental process. Likewise with SED9, and that's normally functioning, it prevents excess apoptosis. But when you mutate SED9, it doesn't work properly. It can't prevent apoptosis, and now you get extra apoptotic cells during embryogenesis. So what are these? Let's start with SED3. What was it? Well, finally, when they were able to identify the gene and sequencing it, they were able to ascertain that it was a kind of a protease, which they refer to as cysteine-dependent aspartate-derived protease, which is a mouthful, so they call them caspases. And these caspases work by cleaving other proteins. And when they do so, they can, for example, caspases can cleave proteins that are involved in, for example, cytoskeletal structure. So when these are cleaved, they no longer function in their normal capacity to maintain cellular structure. So remember, the cell is destroying itself. You need to sort of break all the support beams and everything that's holding up the cell. So you're going to cleave that cytoskeletal network to cause the cell to break apart. The nucleus undergoes dramatic changes, right? So you get compaction of the chromatin, and the nucleus itself compartmentalizes into smaller pieces. So nuclear lamins control the structure of the nucleus. Those get degraded. And obviously other parts of the cell that we're no longer interested in maintaining the cell, so we get rid of the Golgi apparatus as well. And then finally, there's an inhibitor of DNAs. So remember that DNAs I told you about, that concept, those internucleosomal regions, is very typical of apoptosis. That DNase is held in check by a protein called ICAT, or inhibitor of DNase. And so when the caspase cleave that, it allows the DNase to be freely active and to start cutting at those internucleosomal regions. So the other important aspect is to note that there's two types of caspases. There's an initiator caspase. So these are the ones that are sort of at the top of the chain. They start the process. They are initiated by dimers. And when we talk two proteins stick together, they form a dimer. And so when they're dimerized, that activates these caspases, and then they go forward to cleave and activate other caspases, which we call effector or executioner caspases. These are the caspases that are doing this kind of a dirty work, right? So the cleavage of these caspases by the initiator caspase actually leads to their activation. So they have a negative regulatory domain that gets cleaved off, and that activates the executioner caspases. And then they go on to cleave other protein substrates within the cell to trigger apoptosis. So we have initiators, they get activated by dimerization, they're not cleaved. And when we have the executioner caspases, they get activated by the initiator caspases, cleaving them and activating them. They then go on, the executioner caspases go on to cleave. This is just a sort of general category of proteins that will cause all those morphological changes that I talked about. Okay, so that, so SED3, we'll get back to, a little bit more detail about SED3. So we talked about those SED mutations, SED3 and SED4, that when mutated, you don't get apoptosis occurring. SED3 is a caspase. We'll talk about exactly what type of caspase in the pathway in just a minute. SED4, it turns out that this one, and the other thing that's important to note is that although these proteins and genes were identified in C. elegans, they're conserved all the way up to humans. So, SED4 was the other gene identified in C. elegans that when mutated, you're not getting those apoptotic, you're not allowing apoptosis to occur properly during embryogenesis and work. What does it look like in mammals? Well, it resembles a protein called APAF1, which is apoptotic protease activating factor. And this thing forms this massive structure called the apoptosome, or the 1.4 megadalton wheel of death. It's like a monster truck, you'd say this. With more gusto if I wasn't feeling so crappy. So, this thing will assemble into a 7-armed beast called the apoptosome. So, each one of these little things is kind of shown here. That's APAF1. So, it self-assembles into this large structure. However, it's not activated. It is activated by the release of cytochrome C from the mitochondria, which seems kind of strange because cytochrome C is involved in electron transport chain activity. It plays a really critical role during oxidative phosphorylation. There's another role it plays, is that during the apoptotic process, it gets released from the intermembrane space. So remember, there's two membranes in the mitochondria, and in the intermembrane space, you can have free cytochrome C. It's released through a channel, which we're going to talk about in a little bit more detail as we move along. It enters into the cytosol, and then it binds one cytochrome C binds to each arm of the apoptosome. So each APAF subunit, there's 7, 1, 2, 3, 4, 5, 6, 7, is going to bind 7 cytochrome C. And that acts as a trigger to cause the recruitment of caspase 9. So this is where the SED3, remember, so unfortunately there's a little bit of we do flip back and forth a little bit. The fundamental discoveries were found in C. elegans, and that's all these SED genes. Their equivalents were found in mammals, which tend to have different names. So SED3 is the equivalent of caspase 9 in mammals. Caspase 9 is an initiator caspase. It gets activated by dimerization, but what promotes its dimerization is the activation of the apoptosome. So that big APAF 7-spoke wheel, each end of the wheel and the spoke is a cytochrome C bound, and that acts as a trigger to cause recruitment of 2 caspase 9s sort of into the underside. Imagine it's like a little, I don't know, a squid or something. And it's where my palm of my hand is. That's where the 2 caspase 9 subunits will dimerize. And that leads to the activation of caspase 9, which will go on and cleave other caspases or execution of caspases to initiate apoptosis. All right. So, and I'll come back to this again. We'll go through these processes, but I'm going to add a little bit more detail as we move along. So we talked about SED3, we talked about SED4. Those are things that help promote apoptosis. SED3 related to caspase, SED4 related to the APAF, part of the apoptosome. What are the other kind of regulatory members of apoptosis? And surprisingly, this gene, the BCL2 family, we're actually identifying in humans first. In part by studying cancer, in this case a B-cell lymphoma. So it's a blood cancer in which the blood lymphocytes grow uncontrollably. And they found that there was an amplification of a gene, which they called BCL2, B-cell lymphoma 2. And so when we look at BCL2, it has highly conserved domains called BCL2 homology domains, or BH domains. So we have 1, 2, 3, 4, and then there's also a transmembrane domain, TM. So BCL2 was the first one that was found, and then we found, there's actually a lot of other genes that encode for very similar type proteins, look a lot like BCL2. And then we've got things like BCL, XL, and these other gobbledygook ones, which I'm not going to focus on. not really important. So these ones are actually suppress apoptosis. They're pro-survival proteins. They found, though, that there's other proteins that look very similar to the BCL2 protein, but they may be missing one of the BH domains. So in this case, these proteins have 3 of the BH domains, but they're missing the 4th one. They also have the transmembrane domain, and these are pro-apoptotic members. So we'll talk about BACs and BAC in a minute. And then finally, there's one other BCL2 family member, and these are the BH3 only family of proteins, and hence their name. They only have one BH3 domain. And so we'll talk a little bit about a protein called BID in a minute. So these guys promote apoptosis, these guys suppress apoptosis. And these ones up here are capable of oligomorphism, so they stick together. So what does this have to do with apoptosis? Well, we'll get there in a minute. So at this point, I'm just going to reiterate some of the players I talked about. We've talked a little bit about this part of the pathway where we have release of cytochrome C from the mitochondria, and that's going to bind to APAP1. It's going to trigger the formation of the apoptosome, and that in turn will cause caspase 9 to dimerize with this structure, activating it. So it's going to activate effector or execution of caspases like caspase 3 or 7, which will go on to cleave various cellular targets. I'm not going to query you about this. There's many different ways of activating apoptosis. I'm just focusing on the classical pathway, so we don't need to work about SMAC, Diablo, and XI, AP, or whatever the hell it is. Doesn't matter. Now, we talked about these BCL2 proteins. So as it turns out, we can have these BH3 only proteins, which do not associate with membranes. They're kind of floating around in the cytosol. And they will bind to and inhibit BCL2. Now, BCL2 is actually an inhibitor of these other proteins, BACs and BAC. And I'll explain in a little bit of detail what does it mean by inhibiting. These can integrate into the outer mitochondrial membrane and form an oligomeric pore, which will promote the release of cytochrome C. And BCL2 prevents that. So it's a competition. These guys are always competing with one another. These guys, BH3 and the BAC and BAC, are always trying to promote apoptosis in slightly different ways, while BCL2 is trying to put their brake on it. So this is the mammalian side of it. This is the C. elegans side in nematodes. So SED9 is the equivalent, it would be BCL2. SED4, the equivalent would be APAP1. And SED3, the equivalent would be caspase 9. I'm not going to worry about this. Just more acronyms, already too many. Okay, so now let's look at a little bit now, try to put it together in like maybe a more physiological context. I talked about trophic factor withdrawal. I mentioned this as one of the early signal transduction lectures. We talk about signals. We can have signals that can cause cells to grow and divide. We can also have signals that tell us to survive. So these are trophic factors. So they can bind to a thing like an RTK, which we talked about last lecture. That can lead to phosphorylation cascade, which in this case is a kinase called PA3 kinase. It'll phosphorylate a protein called protein kinase B, and that in turn phosphorylates a BH3 only protein called BAD. Now, when BAD is phosphorylated, it can be recognized by an inhibitory protein called 1433. We talked about that a little bit when we talked about RAF inhibition. So 1433 binds to phosphorylated protein and is generally involved in inhibiting them being active. So when we have our trophic factor present, we're getting this kinase pathway activated. It's phosphorylated BAD, and that in turn allows it to be recognized by 1433. Nothing's happening. We remove the trophic factor. This kinase pathway no longer is active. BAD is no longer phosphorylated. And the unphosphorylated version of BAD can interact with BCL2. Now, it will inhibit BCL2. What is BCL2 normally doing if it doesn't have any inhibition? It's binding to these other protein members. Remember, they have the 3 BH domains. They're lacking the 4th one. So BAC or BACs. And so it binds to and prevents these proteins from oligomerizing. So they stick together and they form a channel. And when they form that channel, this allows the release of cytochrome C from the intermembrane space into the cytosol. And just as we talked about previously, cytochrome C combined to APAF1 and apoptosome, and activated caspases, etc., etc. So the key thing to take note of is BCL2 is binding to and inhibiting these pro-apoptotic proteins BAC, BAK, or BAX, BAX. All right, so if that's happening, no apoptosis. But if we no longer have this phosphorylation cascade occurring because the trophic factor is not present in that cell, that is no longer phosphorylated, moves over to the outer mitochondrial membrane and binds onto BCL2 and stops BCL2 from now binding and inhibiting BAC or BAX, allowing it to oligomerize and then allowing cytochrome C to be released into the cytosol. Okay. So that is an example of what we would call the intrinsic pathway. You're simply removing a survival signal, and by default, the cells will allow this to happen, the oligomerization of BAC and BAX. And so this is predominantly driven by the mitochondria in the release of cytochrome C. What about the extrinsic pathway? So this might be involved, for example, in an immune cell, recognizing there's a cancer cell, because cancer cells quite frequently produce certain cell surface proteins that normally shouldn't be produced. And so this can trigger the association of an immune cell to a cancer cell. And in this case, the signaling of the immune cell can produce what we call a death signal. It could be something like tumor necrosis factor or fast ligand. And it'll bind to these, so this is our death signal, so it's either TNF-alpha or fast ligand. And let's say this is the cancer cell, it will be expressing a death receptor, either the TNF-alpha receptor or the fast ligand receptor. And that, in turn, causes activation of this receptor with some other stuff, which I don't really care about. Ultimately, the activation of the death receptor will activate an initiator caspase. Let me step back for a second. Will take procaspase 8 and turn it into, an initiator caspase 8, and it promotes its dimerization, which isn't actually shown here. So the dimerized caspase 8 can go on to cleave effector or executioner caspases like 3, 6, and 9, and then they're going to go on and trigger programmed cell death. Now, there's another arm of this pathway that's activated, and that's the cleavage of another BH3 domain called BID. And so BID exists in a longer form, and it's generally benign, but when it's cleaved by caspase 8 to form TBID, it's a smaller protein, and it can go down and it can bind to BCL2 and inhibit it. Very similar to that. It's a little bit different in how it's activated. It's activated by cleavage by caspase 8, but it does the same kind of thing in that it will go to and bind to and inhibit BCL2 at the mitochondrial outer membrane. And BCL2 can no longer inhibit. This is a mistake in the textbook. It is not bad. It is BAK, B-A-K. BACs, they got that right. So by TBID binding to BCL2, BCL2 now can no longer prevent the oligomerization of BAK or BACs, and then it forms this oligomer, and then it allows cytochrome C to be released, activation of the apoptosome, activation of caspase 9. So in this case, you're getting two ways of activating apoptosis. A fairly quick one, by taking the initiator caspase 8 and allowing it to dimerize very quickly, and then immediately activates those execution of caspases. And it also, through a couple more steps, will cleave BID to TBID, and that's going to inhibit the BCL2 arm of this pro-survival mechanism, allowing the oligomerization of those pro-apoptotic proteins. And so you're getting a 1-2 punch. So this can be a very effective way of initiating apoptosis very quickly and robustly. If it's a cancer cell, that's probably a good thing. Okay, so I'm just reiterating things that we talked about. We talked about 2 ways of initiating apoptosis. So the intrinsic pathway through trophic factor withdrawal. That's a thing like an RTK that's no longer activated, can no longer phosphorylate and inhibit BAD, for example. Those BH3 only proteins normally when activated. So if it's BAD, it's going to be through no phosphorylation. BID would be activated through the extrinsic pathway. A DEF signal activates caspase 8, which cleaves BID to TBID. And unphosphorylated BAD or TBID both work by binding and inhibiting BCL2. BCL2 can no longer inhibit BAC, and so those proteins can oligomerize at the outer mitochondrial membrane. That can cause the release of cytochrome C into the cytosol, going to bind to APAF1, big 7-wheel megadol thingamabob, and that in turn causes a recruitment of caspase 9. 2, they dimerize. The dimerization of caspase 9 causes its activation, and it cleaves and activates these downstream effector or execution pathways, like 3, such or 6 or 7. And then they go on and cleave things like ICAD and cytoskeletal stuff and nucleolanin, et cetera, et cetera. So that's basically the bare bones of apoptosis. It is way, way more complicated than that, but this is sort of the historical early things that we discovered. We now know there's many, many other ways to regulate it, but these are sort of the main players. Okay, so how does this have relevance? Okay, so developmental processes, obviously it's important. It's important for maintaining cellular homeostasis. Old, worn out cells, we need to get rid of them. Or cells that incur mutations that could make them potentially cancerous. We don't want them lingering. And when we think about cancer, we think about mutations like which we talked about earlier in those signal transduction pathways that cause excessive cell growth. And it could be things like a mutation in RAS or an RTK. So it's kind of those proto-oncogenic mutations that make the cell much more likely to respond either to very little mitogenic signal or even no mitogenic signal. But in reality, that's not very rarely is it the only event that causes cancer. It's usually a couple of events. So it may be with one initiating mutation event which allows some destabilization of the genome, some dysregulation of growth regulatory pathways, which we just talked about in the last lecture. But another arm that tends to be quite prominent in the development of cancer is the cancer cells now start to try to block apoptosis. So it could be mutations within some of these pro-apoptotic BCL genes or some other pro-apoptotic gene. Or it's an augmentation, as we talked about in B-cell lymphoma, extremely high levels of BCL2 being made. So this is actively suppressing apoptosis. So in this case, if we're blocking cells being killed, so we could have a cytotoxic T cell that's producing fast ligand or TNF alpha, and there's been some mutational events which makes them not respond to it or appropriately activate the apoptotic pathway. And as a result, this allows these cells to grow uncontrollably and also resist our body's own defense to try to get rid of them. So one of the research that I am interested in is brain disorders, neurodegenerative diseases like Alzheimer's disease. So this is an example where apoptosis is maybe being activated excessively. And by no means does this summarize everything related to apoptosis in an Alzheimer's brain, but Alzheimer's disease is the most common form of dementia, and it's characterized, particularly in the later stages of the disease, by excessive cell loss, mostly of neurons. If you actually look at the Alzheimer's disease brain compared to a normal brain, it can lose up to a third of the mass. So it's massive cell loss, and it's usually in cell loss, it starts in places that control the formation of new memories, and then it progresses to other parts of the brain that control executive function. So it can be activated in Alzheimer's diseases. There's lots of evidence that there's excessive activation of apoptosis. Some of it can be through trophic factor, inadequate trophic factor signaling. So these trophic factors like brain-derived neurotrophic factor or neurogrowth factor, there's some sort of perturbation, and you're not making those neurotrophic factors. And so this can lead to activation of BAD, which we talked about in the last slide, and that ultimately can lead to apoptosis through the traditional pathways. And then there's also quite a bit of evidence that amyloid beta. So this is a protein, it's a small peptide that's produced in our brain. We produce it all the time. We have lots of it in our brain. But it tends to accumulate more so as we get older. And in some individuals, it's processed in a slightly different way. And it's a very promiscuous type of protein in that it can get into various places that you don't want. There's evidence that amyloid beta actually, because of its amphipathic nature, it can cross membranes. It can enter into the mitochondria and actually causes the mitochondria to become dysfunctional and more likely to depolarize and form those channels to release cytochrome C and promote apoptosis. And I could devote several lectures to that, but this is just to give you a hint the flavor of how this has relevance. So apoptosis, it's good and it's bad. We need it because we need to get rid of unwanted cells. It could be cancer cells or cells that are just no longer serving their purpose anymore, and they can enter into something called senescence. And the senescent cells actually start to produce pro-inflammatory cytokines, which are really not very helpful for various tissues. So we need to get rid of either cancer cells that are growing uncontrollably, or cells that have just lived too long and are worn out, and they're actually now adopting a negative phenotype that are causing problems. So we need to regulate apoptosis. So if we have too much, it could be a situation where like a degenerative disease like Alzheimer's disease, or even inflammation of joints and whatnot can cause deterioration of cartilage. So anything where we're excessively getting rid of cells that we need is bad. Conversely, if we don't have enough apoptosis and we're not getting rid of undesirable things like cancer cells or senescent cells, that can also be problematic. So, again, too little apoptosis, where can that happen and where can that be problematic? Well, I have webbed toes, it hasn't really hurt me, but there are some more serious developmental disorders where by not activating apoptosis, you end up with other tissues that form inappropriately and cause pretty serious developmental issues. Not being able to get rid of cancer cells is obviously also a big problem. Having immune cells that can't activate apoptosis appropriately and allowing things like infectious agents to persist because we can't trigger the death of maybe an infected cell. Too much apoptosis, we talk about degenerative diseases like Alzheimer's and Parkinson's disease. Ischemia reperfusion, that's a very common way where there's too much apoptosis. In this case, it's a blockage of blood flow to one part, maybe it's the brain or some other type of tissue. And that in of itself isn't super harmful. It's if the clot releases and you get the inflow of fresh oxygenated blood, it rapidly jumpstarts all the mitochondria. And when mitochondria go from really low key to super active, they're much more likely to release cytochrome C and trigger apoptosis. We have a number of autoimmune diseases that, again, are triggering the death of healthy cells. And then finally, any of you actually are interested, I teach a fourth-year course on aging and the apoptosis of stem cells and many other types of cells in our body, particularly stem cells, are believed to be one of the major contributing factors to aging itself. So with that, that completes the course. And I'll just make a few just brief announcements that we're going to post some information about the final exam. It's going to be 50 multiple choice questions. It will be cumulative. With that being said, the content is going to be biased towards what you'll be tested on more so is the content from test 4 on. So everything I've covered in my lectures, last 4 lectures, and the last couple of lectures that Dr. Damianowski delivered. However, there may be some material from the very first lecture. It's all testable. So keep that in mind. I would suggest if you're pressed for time, study hard on the latter part of the material, but do go over your notes and look at some of the early parts just to refresh your memory. And with that, good luck.